Hey guys, this time I come with a video from biology and this is the exercise 6. This is basically the NCRT lab manual and we are going to do the preparation and study of mitosis in onion root tips, which is our aim. What is the principle? See, the somatic growth in plants and animals also takes place by the increase in the number of cells, right? A cell divides mitotically to form two daughter cells wherein the number of chromosomes it remains the same, that is they are unchanged as in the mother cell. So in plants, such divisions they rapidly take place in the meristematic tissues of root and shoot apexes, where the stages of mitosis can be easily observed. In animals, mitotically dividing cells they can be easily viewed in the bone marrow tissue of a vertebrate, epithelial cell from the gills in the fishes, and the tail of growing tadpole larva of frog. Okay, so what are the requirements? Onion bulbs, wide mouth glass tubers, jar, bottle, glacial acetic acid. This is an important constituent. Ethanol, 2 to 4%, acetocarmine or acetoocean stain, um, normality by 10 HCl, spirit lamp, hot plate, slide, cover slips, blotting paper, molten wax, nail polish, and compound microscope. So, what is the procedure? Growing of root tips. See, select a few medium-sized onion bulbs and carefully remove the dry roots present. The Grow the root tips by placing the bulbs on a glass tuber for about 3 to 4 centimeter diameter filled with water. So care should be taken so that the stem portion of the bulb, the basal part, just touches the water. Now, a few drops of water may be added periodically to compensate the evaporation losses. New roots may take 3 to 6 days to grow. Now, you have to cut 2 to 3 centimeter long freshly grown roots and transfer them to freshly prepared fixative, that is acetoalcohol 1:3 glacial acetic acid with ethanol. Keep the root tips in fixative for 24 hours and then transfer them to 70% ethanol for preservation and use in future. Now onion root tip have a cell cycle of approximately 24-hour duration, that is they divide once in 24 hours, and this division usually takes place about 2 hours after sunrise. Therefore, the roots grown on water should be cut only at that time to score maximum number of dividing cells. Okay, so the preparation of slides. Take one or two preserved roots and wash them in water on a clean and grease-free slide. Now place one drop of N by 10 HCl on the root tip, followed by 2 to 3 drops of acetocarmine or acetoocean stain on it. Now leave the slide for 5 to 10 minutes on a hot plate or warm it slightly on a spirit lamp. Care should be taken that the stain is not dried up. Carefully blot the excess stain using the blotting paper. Now you have to cut the comparatively more stained tip portion of the root and retain it on the slide and discard the remaining portion. After 10 to 20 seconds, put 1 or 2 drops of water and blot them carefully using the blotting paper. Again, you have to put a drop of water on the root tip and mount a cover slip on it, avoiding air bubbles. So place the slide in between the folds of blotting paper using the fingers in such a way that the cover slip mounted on the slide is properly held. Now slowly tap the cover slip using the blunt end of the pencil so that the meristematic tissue of the root tip below the cover slip is properly squashed and spread as a thin layer of cells. Now carefully seal the margins of the cover slip using molten paraffin wax or nail polish. This preparation of onion root tip cells is now ready for the study of mitosis. Okay. So, how we have to study? See, place the slide on the stage of a quality compound microscope. Now, first observe it under the lower magnification to search for the area having a few dividing cells. Examine the dividing cells under high magnification of the microscope to observe the detailed features of the mitosis. What is the observation that you get? The stages of mitosis can be broadly categorized into two parts: karyokinesis, which is the division of nucleus, followed by cytokinesis, which is the division of cytoplasm and ultimately of the cell. Those cells which are not in the phases of cell division, they are considered to be in interphase. You may observe that most of the cells in a microscope field are in interphase. The cells are mostly rectangular, oval, or even circular in shape. with almost centrally situated, densely stained nucleus. The chromatic or colored material of the nucleus is homogeneous and looks granular. The boundary of the nucleus is distinct. One or few nucleoli can also be observed inside the nucleus. Now, what is the stage of mitosis? Prophase. Intact nuclear outline is seen. The chromatin, which is seen as homogeneous material in the nucleus at interface, appears as a network of fine threads or chromosomes. Nuclei may or may not be visible. So this is how you see the interface in under the microscope. Okay, the next is prophase. Okay, the prophase is not yet over. If the cell under observation is in the early stage of prophase, then the chromatin fiber are very thin. However, in the cells at late prophase comparatively थोड़े से thick हो जाते हैं chromatin fibers. Okay, besides this, in the late prophase जो nuclear membrane है, it may not be noticeable. It may not be noticed. मेटाफेज, द न्यूक्लियर मेम्ब्रेन डिसअपियर्स. मेटाफेज आते ही तो न्यूक्लियर मेम्ब्रेन आपको नजर ही नहीं आएगी. क्रोमोसोम्स थिक हो जाएंगे. दे आर अरेंज एट द इक्वेटोरियल प्लेन ऑफ द सेल एंड ईच क्रोमोसोम at this stage has two chromatids which are joined together at the centromere, which can be seen by changing the resolution of the microscope. न्यूक्लियोलस जो है अब आपको नहीं नजर आएगा. एनाफेज में क्या होता है, सेपरेट हो जाएंगे क्रोमेटिड्स जो हैं हर एक क्रोमोसोम के. द क्रोमेटिड्स सेपरेट ड्यू टू द स्प्लिटिंग ऑफ द सेंट्रोमेयर, राइट. Each chromatid now represents a separate chromosome as it has its own centromere. The chromosomes are found as if they have moved towards the two poles of the cell. The chromosome at this stage may look like the shape of alphabet V, J or I depending upon the position of the centromere in them. So different anaphase cells show different stages of movement of the chromosome to opposite poles. Okay, and they are designed to represent early, mid and late anaphase. So telophase, chromosome reach the opposite poles, lose their individuality and look like a mass of chromatin. Nuclear membrane appears to form the nuclei of the two future daughter cells. What is cytokinesis in the plants? Okay, so before starting this, we shall see these pictures. The first one was the interface. In the interface, this is something like this. You can compare it with this diagram. First of all, see these figures that it was actually like this in its prophase. Means in its interface, it was about to divide. As soon as the prophase came, you can see the nuclear membrane and the chromosomes, they have become a bit thick. When it arrives to metaphase, it is very thick and you can see that the spindle have been separated and the metaplate at the metaplate the chromosomes have arrived. Now this is the anaphase separation and finally at the telophase. If we compare it with the actual diagrams, this is very means representing it. If they are very light, the stains are very light, you can say that it is interphase. It has not divided yet. If the stains are a bit darker but they are intact, they have not separated yet, then you can say that prophase is going on. If they have started to separate from each other, you can say the metaphase is on. And if you are seeing them away from each other, you can say that the anaphase is going on. And if they are, in one cell you are seeing two, you can say that it is the telophase. Okay, so these are the stages of mitosis. Actual microscopic view on the left side and its diagrammatic representation on the right side. So now we were on the second part, which was the cytokinesis. So in plants, a cell plate is formed in the middle after the telophase and the plate can be seen to extend outwards to ultimately reach the margin of the cell and divide the cell into two. So such cell plates are characteristic of plant cells. However, in an animal cell, the two sides of the cell show empushings or constrictions formed from the peripheral region in the middle of the cell. I repeat, जो ऐसे सेल प्लेट्स हैं ये प्लांट सेल की कैरेक्टरिस्टिक प्रॉपर्टी है, जबकि एनिमल सेल के अंदर द टू साइड्स ऑफ द सेल दे शो इम्पुशिंग्स, मतलब बाहर से अंदर आती हैं. ओके, so draw the label diagram of all the phases of mitosis. Now what is the discussion? Mitotic index mi is defined as the ratio of the total number of dividing cells and the total number of cells in a particular focus chosen randomly under the microscope and is calculated as microscopic index is equals to small n, that is total number of dividing cells, upon capital N, that is the total number of cells into 100. So by randomly selecting 5 to 10 such foci, one can estimate the mitotic index of for a given type. The effect of different samples of water, whether it is polluted or contaminated, can be assayed on the mitotic index, an inductive feature of somatic growth rate in them. Further, the impact of foreign type of pollutants on different phases of the mitosis can also be assayed. Tabulatory observations in the tabular form given below. So the questions are, you have to suggest names of a few tissues which are suitable for the study of mitosis. Why is mitosis also known as equational division? What shape would a metacentric and a submetacentric chromosome exhibit during the anaphase stage? And how does cytokinesis differ in plants and animal cells? So this was all about our experiment number 6 and our exercise number 6, and I hope you like this video. I hope it helped you a lot, and thank you so much for watching this video.